Hello there everyone, welcome to our first topic for the midterms. A discussion of the inheritance and formation of ABO antigens requires an understanding of the H antigen, which is inherited independently of the ABO blood group system antigens. The H gene genetically controls the production of H antigen, which is located on a different chromosome from the ABO genetic locus. In addition to the ABO and H genes, the expression of soluble ABO antigens is influenced by inheritance of the secretor gene. The secretor gene genetically influences the formation of ABO antigens in the saliva, tears, and other body fluids. Consequently, occurrence and location of the ABO antigens are influenced by the three genetically independent loci, ABO, H, and secretor gene. At the end of the week, the students are able to describe how to determine the patient's secretor status through saliva testing, enumerate and prepare equipment and materials needed in the determination of secretor status, identify the ABO substances found in the saliva, and explain the principle of the method for secretor status determination. Even though it is not routinely performed in the laboratory, however, secretor status determination is vital in order to confirm ABO phenotype of an individual, particularly if they have ABO discrepancies in blood typing, as well as if an individual has either the weak A or weak B subgroups. This is also used to categorize a Bombay phenotype individual if they belong to category 1, 2, or 3 if they have ABH substances in their secretions. There are two allelic genes at this locus, the secretor gene and the amorph gene, or the capital SE gene and the small SE gene. So the gene product of the secretor allele is the fucosyltransferase 2, which is an L-fucosyltransferase that preferentially adds L-fucose to type 1 oligosaccharide chain structures in the secretory glands. So the fucosyltransferase 2 gene may also act on type 2 chains in the secretory glands. The H gene, fucosyltransferase 1, preferentially adds fucose to type 2 chains. The fucosyltransferase 2 gene is directly responsible for regulating the expression of soluble A, B, and H antigens on the glycoprotein structures located in body secretions such as the saliva. Now let us define what is a secretor and a non-secretor. Secretor is an individual who inherits the capital SE allele and express soluble forms of H antigens in the secretions, while non-secretor on the other hand is an individual who inherits the genotype amorph or the small SE and does not express soluble H substance in the secretions. Therefore, an individual who inherits the capital SE allele in their either a homozygous capital SE or a heterozygous capital SE small SE manner is classified as a secretor. About 80% of the random population inherits the capital SE allele. These individuals are classified as secretors. So these individuals express soluble forms of each antigens in their secretions that can be converted to A or B antigens by the A and B glycosyltransferases. These soluble antigens are found in the saliva, urine, bile, amniotic fluid, breast milk, exudate, and digestive fluids. So an individual with the genotype amorph SE is classified as a non-secretor with a frequency of about 20% of the random population. The allele small SE is an amorph. A homozygous does not convert glycoprotein antigen precursors to soluble H substance and has neither soluble H antigens nor soluble A or B antigens present in body fluids. Let's just review the structure on the formation of our ABH antigens. So the common structure or antigen building block for A, B, and H antigens is an oligosaccharide chain attached to either a protein or a lipid carrier molecule. The oligosaccharide chain comprises 4 sugar molecules linked in simple linear forms or complex branch structures. The two terminal sugars, D-galactose and N-acetylglucosamine, may be linked together in two different configurations. So when the number 1 carbon of your D-galactose is linked with the number 3 carbon of N-acetylglucosamine, the linkage is described as beta-1,3. Type 1 oligosaccharide chains are formed. So when the number 1 carbon of D-galactose is linked with the number 4 carbon of N-acetylglucosamine, the linkage is described as beta-1,4. So type 2 oligosaccharide chains are formed. Type 2 structures are associated primarily with glycolipids and glycoproteins on the red cell membrane, and type 1 structures are associated primarily with body fluids. Some type 2 glycoprotein structures are located in body fluids and secretions. So in this table, you can see the various genes, red cell antigens, and substances which is found in the saliva. Antigens that are expressed on the red cells and in the secretions varies depending on the set of antigens that an individual has inherited. If an individual has inherited the homozygous A gene, heterozygous H gene, and homozygous secretor gene, the paragloboside in the red cell and secretions is converted into H antigens. Therefore, A and H antigens are expressed both in the saliva and in the red cell. So if an individual has inherited the heterozygous B gene, the homozygous H gene, and homozygous or amorph secretor gene, so the paragloboside in the red cell and in secretions is converted into H antigens. Therefore, B and H antigens are expressed. But because you're a non-secretor, then there's no expression of B and H substances in the secretion. If an individual has inherited the homozygous O gene, heterozygous H gene, and heterozygous secretor gene, remember that O is an amorph gene, so the paragloboside in the red cell and secretions is converted into H antigen, therefore H antigen is expressed. If an individual has inherited the homozygous O gene, homozygous amorph H gene, and homozygous amorph secretor gene, therefore, no antigens will be expressed in the red cell and secretions. The individual can also be referred as the classical Bombay phenotype. Now let's proceed to the method used for determining the secretor status of an individual. Heme agglutination inhibition reaction is also known as your neutralization reaction. So before blood transfusion, ABO and RH blood typing is performed to check the compatibility between the donors and recipients' blood types. Besides pathogens, non-self blood types can also induce antibody production. To test the donors and recipients' blood types, recombinant antibodies developed against AB, O, and RH antigens are mixed with donors and recipients' red blood cells, and heme agglutination is determined. In simple words, an A-positive blood sample will agglutinate with anti-A and anti-RH antibodies. So a cross-match analysis is also carried out after the determination of blood type. In this case, the donor's red blood cells are incubated with the recipient's serum. In the recipient's serum, the presence of antibodies against the donor's red blood cells will result in heme agglutination. Now let's discuss the principle of heme agglutination inhibition reaction. Heme agglutination occurs when the antibody and the red blood cells are mixed. But if the serum of an individual infected with the antigen and is mixed with the RBC and the antigen, there won't be any agglutination of your red blood cells. So this phenomenon is known as heme agglutination inhibition. This arises because antibodies present in the serum of that infected individual will react with the antigen and neutralize them, which yields a positive result. As the heme agglutinins are occupied, so they can't bind and agglutinate RBCs. So from the word itself, inhibition, so therefore, it cannot agglutinate your red blood cells. So if the patient's serum do not contain antibodies against surface proteins of the corresponding antigen, then there will be a heme agglutination reaction as the surface molecules are free to heme agglutinate your red blood cells, and this is a negative result. As for the materials and equipment for the procedure, so we need Wassermann test tubes, Pasteur pipette, beaker, thermometer, test tube rocks, centrifuge, hot plate, Nesco film, and a marking pen. As for the reagents or the samples to be used, so we have unknown saliva samples, 5% red cell suspension of known A, B, and O cells, anti-A serum. So you are gonna make a 1:4 dilution, 1 ml of anti-A sera and 3 ml of NSS. Anti-B serum, you're also going to make a 1:4 dilution, 1 ml of your anti-B anti-sera and 3 ml of your NSS. And then anti-H lectin, 1:4 dilution, so 1 ml of your anti-H and 3 ml of your NSS. And as well as our normal saline solution in a wash bottle. Next is our sample preparation. So first, you have to collect approximately 2 ml of saliva in a small beaker, and you have to transfer it into a Wassermann test tube. So be sure to cover it with Nesco film or a parafilm. So place the test tube with saliva in a boiling water bath for 10 minutes, and please take note that heating the specimen will inactivate the enzymes present in the saliva. However, when done excessively, it might destroy blood group substances and give a false negative result. Then next is you have to centrifuge your Wassermann test tubes at 3,400 RPM for 10 minutes. And then transfer the clear or the slightly opalescent supernatant into a clean Wassermann test tube. Then discard any opaque or semi-solid material. So make a 1:2 dilution of the collected supernatant with NSS. So 1 ml of your clear supernatant plus 1 ml of the NSS. As for the anti-sera, you can prepare a 1:4 dilution of your anti-A, anti-B, and anti-H anti-sera. Then you can prepare 5% suspension of your A, B, and O cells. O cells are used because they have the greatest amount of H antigen. And then prepare 3 Wassermann test tubes and label them as A, B, and H respectively. And then next is you can follow this table as your guide. So you have to mix and cover after doing what is being said on the table with your parafilm and or Nesco film. And then after that, you have to incubate the tubes at room temperature for 30 to 60 minutes or for 15 minutes in a 37 degrees Celsius water bath. Then centrifuge for 15 seconds at 3,400 RPM. Next is you have to place 1 drop of sample in each test tube. And then add 1 drop of anti-sera in designated tubes. Next is to mix and cover and then incubate at room temperature for 10 minutes. And then add 1 drop of reagent red cells in designated tubes. Then mix and cover, incubate in 37 degrees Celsius for 15 minutes. Centrifuge for 15 seconds at 3,400 RPM. And lastly, gently dislodge the cell button and observe for agglutination. And you must grade each reaction and record the results. So as for the grading, so you have 0 for no agglutination, 1 plus for medium and small sized agglutinates, 2 plus many medium sized agglutinates, 3 plus several large agglutinates, and 4 plus is a 1 red cell button as a solid agglutinate. For the interpretation of results, so at the table, the plus sign there means agglutination occurred or happens, and then 0 means no agglutination happens. So therefore, if your known A cells has no agglutination reaction, your known B cells has an agglutination reaction or a positive reaction, and your known H cells has no agglutination, so therefore, the soluble substances detected are A and your H antigen, and therefore it is blood group A on your ABO blood type. And then if your tube A has a positive reaction, tube B has no agglutination reaction, tube H has no agglutination reaction, so the soluble substances detected are B and H antigens. Therefore, it belongs to your blood group B or B blood type. And then the next one, if your tube A, tube B, and your tube H has no agglutination at all, therefore the soluble substances detected are A and B and H antigens. So therefore it belongs to your AB blood type. And then if your tube A has positive reaction, tube B has positive reaction, and tube H has no agglutination, the soluble substance detected is your H antigen only, and it belongs to your blood group O. As for the interpretation of results, the absence of agglutination in saliva is a positive result of the presence of ABH antigens and indicates that an individual is a secretor. The diluted reagent antiserum has been neutralized by soluble blood group antigens in the saliva. Hence, no antibodies in the antiserum are free to react with the antigens on the surface of the reagent red cells introduced into the test. The purpose of dilution is to prevent zonal reactions due to either excessive amount of antigen or the post zone or excessive amount of antibody or the pro zone. Excessive levels of soluble ABH substances can be observed in rare cases such as pseudomucinous ovarian cyst, carcinoma of the stomach or pancreas, or intestinal obstruction. So the level of ABH substances in a person may be so high that it inhibits the anti-A or anti-B reagent sera. This interference with ABO blood grouping is due to neutralization of the reagent and anti-A or anti-B by the excess-specific soluble substances, leaving no unbound antibody to react with the person's erythrocytes. So the problem with excessive blood group soluble substances often leads to ABO discrepancies. So this can be resolved through washing the blood sample several times with NSS in order to remove the soluble substances in the plasma. And that ends our lesson for secretor status determination. I hope that you have learned something. Stay safe and thank you so much, everyone.